Hello, welcome in this hematological section of human physiology. Today's topic is functions of plasma protein. In previous video, I have talked about plasma protein functions in short and what are the types, what are the structures of plasma protein. Today, we are going to elaborate functions of plasma protein. Plasma protein is proteins present in the plasma. Now, what is plasma? In blood, two types form elements and liquid portion. If in a test tube we allow to settle the blood, RBC being heavier will settle in the bottom and upper portion is clear fluid, that is known as plasma. This is clear fluid, it is 45% and it is 55%. In between there is a very teeny layer of WBC and platelets. This is plasma. And plasma has 91% water and 9% solutes, in which this organic portion is plasma protein, and other things are also there, but main is plasma protein. Total plasma protein is 6.4 to 8.3 gram per deciliter of blood. In which there are many types of plasma proteins, but main plasma proteins are only three. They are albumin, globulin, fibrinogen. Albumin is 3 to 5 gram per deciliter of blood. Average is 4.8 gram per deciliter. Globulin range is 2 to 3 gram per deciliter. Here it is 2.3 gram per deciliter. Fibrinogen is 0.2 to 0.45 gram per deciliter, in which 0.3 gram per deciliter is the average. Other are also there. They are prothrombin, transferrin, celluloplasmin, etc. They are present, but they are in very little amount, but of importance. Now, functions. Concentrating on functions of the plasma protein. This maintains colloidal osmotic pressure. Colloidal osmotic pressure mainly due to albumin, 80% due to albumin. Osmotic pressure across the capillary wall. This is capillary wall, suppose this is capillary. So osmotic pressure across this capillary wall are of two types, crystalloid and colloidal. Crystalloid, sodium, glucose, etc., which can pass across the capillary walls, and they exert very little amount, negligible amount of pressure, and therefore the main is colloidal osmotic pressure. Colloidal osmotic pressure is by plasma protein. Plasma protein, in which albumin contributes 80% of osmotic pressure. Osmotic pressure exerted by plasma protein is oncotic pressure. Oncotic pressure is osmotic pressure exerted by plasma protein. Now, this is maintained colloidal osmotic pressure. Here, some applied physiology application or clinical application in whole body. Arterial capillaries have arterial end as well as venous end. At arterial end, hydrostatic pressure is more. Hydrostatic pressure is 37 millimeter of mercury at arterial end, and at venous end it reduces to 15 millimeter of mercury. But osmotic pressure is 25 millimeter of mercury. Throughout the capillary, it is 25 millimeter of mercury. I am talking about the normal case. And likewise, in interstitial fluid, fluid surrounding the capillary, there is hydrostatic pressure 2 to 3 millimeter of mercury and osmotic pressure 3 to 4 millimeter of mercury. They cancel each other. So the main forces are at arterial end hydrostatic pressure of 37 and osmotic pressure is 25 millimeter of mercury. That is 12 millimeter of mercury. Hydrostatic pressure tries to push the fluid outside the blood vessel, and osmotic pressure tries to pull the fluid inside. And at this level, this is arterial end. At arterial end, the pressure is more, therefore fluid will go out of the blood vessel. But here it will come out. But at the venous end, what happens? At the venous end, this fluid is sucked in, moves inside the blood vessel due to at venous end hydrostatic pressure is less, 15 millimeter of mercury, but osmotic pressure is 25 millimeter of mercury. Therefore, fluid enters into the blood vessel. At the arterial end, fluid is removed from the blood vessel, from the capillary, and at this portion, that is venous end, it enters into the capillary. A little amount remains in the interstitial fluid, which is removed rapidly from the lymphatics. This force is a Starling force. This is a Starling force. What is a Starling force? It is responsible for filtration and reabsorption of fluid in the capillary. Okay, clear? Applied in cases where osmotic pressure is less, becomes less due to reduced amount of albumin. For example, in cirrhosis, albumin in liver dysfunction, the albumin is not being formed, is less in amount. Therefore, this osmotic pressure will reduce. Or when albumin is being excreted rapidly, removed from the body, for example, nephrotic syndrome, in that case also osmotic pressure reduces, and formation is there, and this excretion is there. Formation is less and excretion is more. Formation is less in, for example, liver disease, dysfunction of or liver or malnutrition, starvation, etc. There is less albumin, plasma protein reduces in amount, and this will cause reduction in this osmotic pressure. When this osmotic pressure reduces, then force removing the fluid is increased, and more fluid will come out into the interstitial space. And at this end also, this hydrostatic pressure is more and osmotic pressure reduces, but so less amount of fluid will enter into the blood vessel, causing accumulation of fluid in the interstitial tissue. That is edema. That is known as, this case is edema. Second thing is when capillary permeability increases. Capillary permeability increases in infection, trauma, etc. Then in that case also fluid will rapidly go into the interstitial space. This is maintenance of colloidal osmotic. Second is maintain the stability of blood. Due to plasma protein, the RBC Rolex formation is reduced and stability is maintained. Then coagulation of blood. Fibrinogen, prothrombin, etc. coagulation factors are there, which helps in coagulation of blood. Then viscosity of blood, 4 to 5 times that of water. Viscosity of blood is 4 to 5 times that of water. And this maintains systemic arterial blood pressure, peripheral resistance. Viscosity of blood is responsible for peripheral resistance, and then it maintains the systemic arterial blood pressure. Buffer action. buffer action, that is pH is 7.4. This plasma protein tries to maintain the pH. This way, it accepts one hydrogen and it will give proton hydrogen ion to this alkaline solution, maintaining the blood pH. This is buffer action. Then immunity. Gamma globulins, immunoglobulins, there are immunoglobulins IgG, IgM, IgD, IgE and IgA. These are immunity to protect infection and protect us from microorganisms invading organisms. Then WBC form triphons and carols from plasma protein. They are essential for formation or nutrition of the cells. Then transport function. Oxygen in dissolved form in transferrin, iron, transferrin transport iron, ceruloplasmin copper, vitamin A, D, E, HDL, LDL, transprotein, cortisone and other hormones, for example thyroxine, albumin and quinolone. Now reservoir of protein. This is plasma protein is a reservoir of protein. In case of emergency, plasma protein will go into the cell, and in another situation when plasma protein reduces, then proteins from the cell will enter into the blood for plasma protein. They are labile, they are interchangeable, and in this case it is called a reservoir of protein. So functions, three main types: the normal value 6.4 to 8.3 gram per deciliter of blood, albumin 3.5 gram per deciliter, 4.8 gram per deciliter is average, globulin 2 to 3 gram per deciliter, average is 2.3 gram per deciliter. Fibrinogen is 0.2 to 0.45 milligram per deciliter. Gram per deciliter. This is average. Other proteins, plasma proteins are present, prothrombin, transferrin and so many. Function, what are the functions? Number one, colloidal osmotic pressure, which is mainly due to albumin, maintain stability of blood, coagulation of blood, viscosity of blood, maintain systemic arterial pressure by peripheral resistance, buffer action, immunity, triphons and chiral formation from plasma protein, transport function and reservoir of protein. These are the functions of plasma protein. Okay. Thank you for hearing, and if it is of some value, then like it, subscribe my channel and share it. Thank you very much.